So my talk again is a little different, and you heard from Roger Sayer and Dawn Wright and Sean Breyer about the ecological marine units they've developed. So this began a few years ago, and I happened to meet Roger at a meeting like this, and I was very excited about the terrestrial classification he's doing, and I said, oh, we could do the same for the oceans. We have the data, but the computational side's a little difficult. So a few years passed, and then Roger got the collaboration going at Esri, and they'd finished the terrestrial environment, and then they said, let's do the ocean. So that's how this arose. So what I'm going to talk about is try to put that in a wider context and see how it will link to biodiversity, which is my area of main research. So this wouldn't have happened really without the Esri computational skills, the methodologies, and especially some of the visualizations you've seen now at ArcGIS Pro. So why do we want to classify things and do this research anyway? Well, it's kind of fundamental to understanding life on Earth, how it evolved in the past and how it's going to change in the future. More importantly, I've become very interested in the global context, because often when you look at the global context, you understand better the regional and local scale dynamics. And we all love to classify things. We do it in every form of life. It seems to be genetically engineered into us to classify things. It must be the way our brains work. But structural classifications also have heuristic value. They give us information and help us understand things, and they have predictive powers. And when we start looking at some of these classifications, and what happens when we start using data to actually support these classifications? There's three major sources of big data now out there. There's environmental data, which has been pooled together on a website called the Global Marine Environmental Data Sets, where it's all ready to download and use in ArcGIS. We have biological data in the Ocean Biogeographic Information System, which has got over 100,000 species with, I think, 30 million or 40 million species distribution records. And when you get these big data sets, you can now start looking at patterns that in the past people proposed hypotheses based on local and regional scale studies, often on a few different taxonomic groups. And then there's an endless series of papers finding contradictory patterns with other groups in other locations. But once we have all this global data, we can look at patterns. And I've had a few exciting opportunities looking at this. One was in the area of how many species exist on Earth, which I won't go into here. But we found that there was a lot of exaggeration in the literature about it. Another way is recently we just had a paper published in Trends in Ecology where we looked at latitudinal distributions. So the prevailing paradigm in most textbooks about the latitudinal gradient in marine species richness is that it peaks at the equator. You get less species in the polar regions, more at the equator, and certainly more in the tropics. But when we looked at the data, we were quite surprised to find that whether you use the total number of species in a latitudinal band, these are 5-degree latitudes at the bottom, or you use the average number of species, you get this little dip at the equator. So based on looking at that data set of 65,000 species, we went back and looked through the literature, and every single study we looked at, was it mammals, plankton, benthos, any groups, they all had a little dip at the equator, but people just drew a line right through it, because you know that biodiversity peaks at the equator, so you just draw a line through. So that was quite exciting. And who knows what other opportunities exist there to actually challenge some of the prevailing paradigms that we don't even expect. We didn't even go looking for this. It was kind of an accident. So lots of people have classified things for fisheries, nature conservation reporting, and many other purposes. This is the Global Marine Environmental Data Sets, and I'll give you some examples of some of the data patterns there, which help introduce how we then interpret the ecological marine units later. So the ocean is the longest inhabited part of our planet and the largest habitat. We do have some ridges in the oceans, but if we look at the slopes of these, it's really all the slopes are around the coastal areas, which is the red patterns here. You get little islands stretching out into the oceans. So if, say, most species occurred in shallow water, well, then you'd expect most species to be where these red colors are on the map here, around the coasts and in the large continental shelves here. If we look at salinity, we first look at these maps which you find in textbooks, and they typically show patterns of salinity variation. But when you look at the scale, it goes from 30 to 40, because in most of the ocean, salinity is more or less the same, and it doesn't matter to marine species where this salinity variation is irrelevant biologically, even though it's valuable from a physical oceanographic point of view. So the areas with low salinity are really the small coastal areas and some of the inland seas like the Black Sea, the Caspian Sea, and the Baltic Sea. If we look at annual temperature, we can see the strong latitudinal gradient when you look at surface temperatures. When you look at the seabed, you get a completely different pattern of temperature. With warmer temperatures in some of the narrow seas and coastal areas and the shallow areas, and basically most of the deep sea, the same temperature, which is rather like your fridge at home. It's around 2 to 4 degrees. Well, how your fridge should be, I guess. If we look at patterns of nutrients, they're also not homogeneous in the oceans. And we have these strong areas down here, high nitrates. So as all gardeners and farmers know, you need nitrogen and phosphorus to grow things. And if you've got diatoms, they like silicate as well. So you've got actually excess nutrients in the Southern Ocean, both in nitrate and phosphate. But if you look at the deep seabed bottom, you get a completely different pattern again. So there's definitely some different patterns with depth. And you also get these patches of high nutrients up here. So that's kind of surprising when you look at it for first. Why would that be the case? And then the biological response to all these patterns is you start looking at the productivity. This is derived from ocean color data, which is matched to ground truth chlorophyll. And you see that most productivity is actually in coastal areas. And usually when people show these maps, if they're from the northern hemisphere, they always show the northern summer. If you're down in New Zealand or Australia like we are, we always show the southern summer, so you see highest productivity in the polar regions. But actually, when you look at the average over the year, most productivity is in coastal areas and in the tropics, just like it is on land. So biodiversity has a number of components. It's the variety of life. You can be measured at populations, species, and it also includes ecosystems. So 2 ways of measuring it at a global level would be the number of species, the richness patterns like I showed you with the latitudinal gradient, or you could look at endemicity, which is how unique species are in different areas. So if you've been to New Zealand or Australia, you'll quickly realize that many of the plants and animals there are completely unique. They have very high endemicity, whereas on big continental land masses, you get lower endemicity, but it's all based on geographic location. But also we want to include ecosystems. So how do we include ecosystems? Well, we can now use the ecological marine units, which include nitrate, phosphate, and silicate, and we can start looking at structure with a combination of the other variables. So the global pattern of species richness, if we look at actual observations that are available in the Ocean Biogeographic Information System, so this was a database I downloaded in and cleaned up a few years ago, and I've calculated an index to try and adjust for sampling effort, but you can see that most species are in the red, is in the tropics and in coastal areas. Another way of looking at that is to look at species ranges. So you take a sample of those data and you plot the likely range of those species, and then you overlay all the ranges, as people always do, often do in GIS, and you get a similar pattern with the darkest color here around the coastal areas and the tropics, and the lowest richness in the deep sea and the open ocean areas. And if you compare the 2 of these, you can see a very common kind of pattern with most species are coastal and most species in the tropics. But that's only the first step. That just looks at richness patterns and predicted richness patterns. To look at endemicity, we do a similarity analysis or cluster analysis, just as the same was done with the ecological marine units, but using different similarity coefficients. So we could take a group of 5-degree latitude longitude cells, because if we use smaller cells, we have too many missing data ones, and if you use bigger cells, it doesn't really give you enough spatial resolution. Then the ecological marine units used environmental data to actually do the clustering. And then what I'm interested in is try to compare the results of the 2 of these, compare the biology and the environmental patterns. So take a very quick jump to the results. This is the results we got from the biogeographic species data analysis, and we ended up with 30 different biological realms. Some of them are quite small, like in some of the Baltic and the Black Sea. We didn't include the Caspian Sea. Some are very large, like the Southern Ocean and the mid-ocean areas. So most of these realms are coastal, and the offshore realms are larger. So you'll still find in literature that there was a paper published only a few months ago which just said most species occur in the deep sea. There's textbooks that say that, which is completely without any evidence, I may say. And why isn't there most species in the deep sea? Well, the environmental data will help us explain that, as I'll show you in a few minutes. But also some of the species distribution helps explain that. So if we take any group of taxa, as far as I've found so far, these are sea pens. I just picked those because they're kind of pretty. And if you take the different genera and you look at their distribution with depth, the species that occur in shallow water have very narrow depth ranges, but the species that occur in deep water have very deep depth ranges. So they occur over a wide depth range. So as you go deeper, the species have a wider distribution both with depth and with space as well. So you end up getting the deep sea appears to be one large habitat for these species, whereas the coastal areas are much more fragmented with more different habitats and smaller species ranges. So when we clustered the environmental data, we used temperature, salinity, oxygen, nitrate, phosphate, and silicate. So we didn't use depth. Depth is an attribute like latitude and longitude. And there was 52 or 53 million data points clustered at a 1/4 degree latitude, longitude, and at different depth intervals. So what's unique about the ecological marine units, and Roger and Dawn kind of mentioned this already, is that it covers all of the oceans, the surface, all the depth, so it's volumetric. It's the first time somebody's done a volumetric analysis at a global scale, and it's based on data analysis. These are the variables, but also the depth zones that were treated weren't equal, so we had we had 5 meter intervals in less than 100 meters, and in the deeper water, we're looking at 100-meter depth intervals. So the volumes varied with depth. This was to reduce the computational demands, but also because the environmental variables vary less as you go deeper. And the other point I should make is that this is limited to a maximum depth of 5,500 meters. And I suppose we all know how deep the oceans are here, so they're over 10,000 meters. So we're really only covering half the depth of the ocean. That's a summary of the methods. So we're using long-term averages, 57-year averages. The data was normalized beforehand, so each variable is given equal weight. And you end up with very different patterns when you look at the sea surface and the seabed, just as we found for some of the individual environmental parameters. When we look at the clusters that emerge, which we now call ecological marine units, these vary with depth, and the darkness here shows that the different number of clusters with depth. So in some areas of the ocean, you get more EMUs with depth, and in some you get less. And if we try to summarize that, less than 500 meters, we've got 30 of the EMUs out of the 37. So they're most of them are actually shallow water and coastal, just like we found with the numbers of species. So we're now finding a common pattern between environmental variation and where you find most species. And this can be scaled with depth, so that it gets darker as you get less light. Roger showed this slide before as well. We can also then group the different, this is depth going down here, which you can't read, so I stuck in big numbers here, showed the 200 meter, 800 meter, and 5,000 meter contours. And you can see that each of the EMUs varies as you go down with depth. And we may have different zones matching the different concepts that are in the ecological literature, as Dawn mentioned too. We can break those down. So if we look at the coastal inland seas, so these only go to 60 kilometers away from the coast. You can see a whole bunch of them here, very complex, rich of what might be called ecosystems, or at least some type of habitat variation. You get less as you go further out from the coast. And as we go into the mid-ocean areas and the offshore open sea, we end up at fewer again. So we've only got 3 down here in the deeper sea. So if I take the number of clusters, or EMUs, it's the same thing. We've got 24 roughly here in shallow water, and as you move down, you've got less and less. So what we're trying to look for is some jumps, as if you're doing a depth profile of a gradient, and see when do you get more or less. So it's still kind of hard to figure out, well, where do you actually have these zones, which probably explains why in the literature you often have different numbers applied to these zones, based on where people have been working. But it does look like there's different zones here, at least environmentally. Whether they're different biologically is another question. I have some examples in a moment that would raise the question whether these environmental differences are actually biologically significant. If we look at the mean value for each EMU, and we cluster them together to see which are the most important in separating the EMU, depth pops out as the very first one. So then I graph these other patterns here against depth in the next few slides. And nitrate and phosphate don't show really any difference in distinguishing EMU, perhaps because they're very highly correlated. So if we look at the variation of variables with depth, I just realized I can look at this big screen here too. We can look at salinity, and there's an odd thing about salinity, is that you end up with low salinity points of around 20 in some of the deep sea. So that surprised me. I don't know if anybody can guess why that is. We'll have a little quiz in a minute. If we look at temperature, we don't see that pattern. It's a bit more predictable. If we look at oxygen, also, it's kind of what we sort of expect. But note the low oxygen values right down the bottom here. 0 oxygen in some deep water areas. The highest oxygens are in shallow water, which we might expect due to plant production and mixing with air. And temperature is always cold in the deep sea, so it's always less than around 10 degrees. So there's a little warm spot here, probably in the Mediterranean. So could this be a mistake in the data? I wonder. So you get you also get all the variation in the shallow water here. So we see these exceptions. And we'll have another look at the data, go back and see where are these points actually located. So if you go online to the Ecological Marine Unit Explorer site, which may be in the Living Atlas now, is it? No? Maybe it's separate? It is? Good. These places are in the Caspian and Black Sea, and if you look at the depth profile, you can see that it does go down to around 23 parts per thousand, and then it goes to basically nearly freshwater in some areas. So this gives you can move back then, look at the statistics and start exploring the information. But the other point I want to make here is that when you look at the EMUs with depth, they actually switch. So they got the same, you got multiple layers as you go down deep. You've actually got, goes from EMUs number 1, then to 14, 15, 14 to 15. So they're kind of hopping about. So you've actually got multiple layers in these waters, which are environmentally can be separated, but it's highly unlikely that they're biologically different. Any species living there would probably have to be adapted to live in all those different environmental conditions. If we look at nutrients, we can also look at the nutrients against depth. We see nice relationships with nitrate increasing with depth. We can see phosphate increases but then levels off, as does silicate. And then if we plot those nutrients against each other, you would expect a very high correlation with phosphate and nitrate, because that's what all the textbooks tell us. But then we find some of these outliers over here with very low phosphate, but very high phosphate, sorry, but low nitrate. And similarly, when you plot nitrate against silicate, you end up at high silicates but low nitrates. Does anyone know, hazard a guess where these locations are? In the ocean? Where would you find these strange conditions? I don't have any prizes. If I had a prize, that yeah. Antarctica is one I thought of, but actually it's the Black and Caspian Seas again, because most marine oceanographic textbooks ignore the Black and Caspian Seas. They consider them as inland seas, a bit like the Great Lakes or some other place. Yeah, but Antarctica is quite different, as you could see in some of the earlier slides. So we can also cluster the similarity of the EMUs. So maybe from a biological point of view, what we will find is that really these differences down here are not significant, and we should be looking just at differences up here. So maybe there's actually fewer marine ecosystems once we can match the EMUs to biology and define the boundaries. Maybe we'll only have a small number of ecosystems and not the 37 defined here. Another way of displaying that dendrogram is to use a multidimensional scaling plot, which is another way of displaying it, and it shows you some of the different groupings. And you can rotate that around a little bit, so you can rotate into a simpler plot here where each number is one of the EMUs. And isn't it amazing, the shape of that? Does anybody guess what it is? It matches an emu. How cool is that, you know? I mean, Really, we spent all our lives hoping for the answer, and it was right there all the time. Yeah, okay. Right. Well, that was one of the most exciting things I discovered preparing this talk. Okay. We can then delve a little deeper into each of these clusters and look at the patterns. And we see these Black Sea and Caspian Sea matches the Arctic, which Chen Shur mentioned in his data analysis, which was independently done as well. So it's kind of curious. We've got the same environments in different parts of the oceans, but it gets even more spectacular. We look at the upper water column, and we see these strong latitudinal effects here. So this is probably the effect of temperature and solar radiation and light. If we look at the mid-water column, we end up with less. We've only got 7 clusters now. We had 13 before. And they're much larger. And then we go deeper again. We end up with only 3 clusters and much larger again. So here this shows that the deep-sea ecosystems have far less environmental variation than coastal ones, and that once a species is adapted to live in any one of these ecosystems, well, then it can just distribute itself right throughout all these big ocean areas, which is why there's less species in the deep sea. Okay. And there's no influence of latitude. As well, in these patterns down here, we're looking at other effects. Okay. When you look a bit closer at these maps, and you can't see this, you'll find little fragments of EMUs scattered amongst the larger ones. So again, this suggests to us that a lot of these differences are probably not biologically significant. So we should stop fragmenting. If we want to have biologically relevant ecosystems, we need to stop fragmenting these EMUs at some point and look at some kind of spatially coherent units. So Dawn showed this as well. It's already flying ahead of me, showing the points. So we've overlaid here the biogeographic realms that we plotted, which are based on species endemicity, with the EMUs. So the EMUs are the colors and the lines of the realms. And some of them are a good match, and some of them are not a good match. Some of them go right through the middle of EMUs. And then I was thinking about this. Well, why would an ecological marine unit necessarily have to match the boundaries? It does for in many cases. You've got environmental gradients and biological gradients seem to coincide. But in other cases, it could be that the ecological marine units are actually a barrier between different assemblages or communities of species. And of course, geographic isolation is important. So areas that are far isolated will have a different biology, regardless if they have exactly the same environmental conditions as somewhere else. So we need to consider these other complicating factors in interpreting the biology of this. So to go into that a little bit more detail, if we looked at some of the biogeographic realms here, numbers 1 to 8, have good matches in terms of spatial overlap with emus, but many of them don't. So in fact, most of them don't. So to conclude, what future research we're going to do? Well, as you can see, we're starting to look at the biological data and see how we would actually use EMUs and existing biological data to define ocean ecosystems. We actually started this project thinking we were going to define ecosystems, and we had a great debate about should we use the word EMUs or not, and so on. But I think we're right now to kind of separate these different concepts, because this is really work in progress. We want to relate the biological data to the ecological marine units in 3 dimensions. So the analysis I did of the global species was just in 2 dimensions. It included deep water and shallow water data, but nobody has yet ever analyzed marine species distributions in 3 dimensions. But now that would be possible by matching species to the ecological marine unit volumes in the oceans and then doing that analysis. And furthermore, finally, what the future holds. So Roger Sayre and Dawn and Esri are now looking into ecological coastal units, because in the coastal zone we have a very complicated mix of terrestrial, freshwater, brackish, and marine species and environments, and it's very hard to map, and it needs a lot closer ground truthing. And they're also now looking into ecological freshwater units. Thank you very much.